Hello everybody, my name is Julia, and I'm going to talk about the work I've done with Naomi Habib and Neil Friedman from the Hebrew University, which aims to cluster and model gene expression time course data. So, a key goal in biological research is to understand the transcriptional response to stimuli, but that goal is rather ambitious, as such response usually contains activation and suppression of hundreds of genes. Each may have its own dynamical response, which is mediated by dozens of transcription factors. And my question is it enough to know just which genes take part in the response? So my answer is no, because we may have genes which have typical early response with fast onset, fast offset times, and high magnitude. We may have late response genes which have later onset times. So it's important to study the dynamics of the response. And when it comes to response dynamics, what do we really want to know? We want to know the onset time, the offset time, the magnitude, but we can't ask the genes that. All we can really ask the genes is what was their expression value at different points in time. And the answer is usually very noisy and partial. And if we have experiments taken over different time courses, it's very hard to compare between them. So how can we go from the noisy expression pattern, to the dynamical parameters of the response? So one way is to fit the data to a mathematical model. And Chechik et al. Proposed such model. Their model was designed to capture the typical two-phase transcriptional response to stimuli, which involves rise from the base level to a peak, and then a fall to a new steady state. And their model had the following parameters. It had three height parameters: the base height, the peak height, and the new base height. Two temporal parameters of the onset and offset times, and two parameters for onset and offset rates. So in total we have 7 parameters, which are exactly the parameters that we wanted. And we learned those parameters by minimizing error function, which is the distance between the function and the measured data at the time points that we have. And we learned that by conjugate gradient descent. So this is great, right? We have a way to go from the noisy expression pattern to the dynamical parameter that we wanted. But this usually isn't the situation. This looks more like it. We usually, since we have 7 parameters in the function, we usually have in time course gene expression data, we usually have less than that time point, so we get overfitting. Since we have too many possible functions that fit the data, we don't know which one to choose. So we choose the one that fits best to the time points that we do have, but it might not fit well to the time points that we don't have, and it might not assess the parameters well. And here I have to add that Chechik et al.'s original model had actually 6 parameters, because they used only one parameter for the onset and offset rates. And we believe that it's more accurate to use 2 different parameters. So this results in overfitting, and we need to find a way to reduce the search space. But before we do that, let's see what else we can do with gene expression data. So we can cluster gene expression data. So here we have, in each row we have the expression pattern of the gene over time, where red is high and green is low. And the underlying assumption here is it's not really true that every gene has its own dynamical pattern, but there are only a few dynamical prototypes that are shared between genes. So our aim is to cluster the genes such that every cluster has a unique dynamical pattern. And there are a lot of algorithms that do that, but they all rely on the distance measure that you choose. And in our case, since we're talking about expression over time, we have to remember that these measurements are, again, noisy and partial, and we may have a different variation in time. We may have a 10-minute interval at one point, or 8-hour interval at another point. So the usual distance measure that we use, like correlation and Euclidean distance, don't fit well to the time course data. So we need to find a new distance measure. So let's recap. We wanted to model the genes, but we had too many possibilities, so we had overfitting, and we wanted to cluster, but we need to find a better distance measure. So what can we do? So suppose for a minute that we do have clusters from somewhere. We can use the clusters and extract dynamical prototypes and use those as prior knowledge for the fitting to reduce the search space. And then once we get the model and the model parameters, we can use those parameters as a metric for the clustering. So what came first, the chicken or the egg? Well, we have to start from somewhere. So we start with an initial guess for the clusters from naive k-means, and we iterate between the clustering and remodeling until we get to a satisfactory result. And we call that our method dynamic modeling and clustering. And to some of you, it may seem a lot of like k-means, which you choose centroids, you guess centroids, and then you cluster according to the centroids, and then you get the centroids from the clusters, and so on. So how exactly do we do that? In order to fit the data according to the priors, we take the clusters, we extract the prototypes, or priors as I will call them from now on. And now, instead of minimizing the error function of just the distance of the function to the data, we also minimize the distance between the parameters of the function, of the fitted function, and the parameters of each prior. So we do that for each prior. And we choose the best one, the one that minimizes the error function, and for each gene we remember which prior it was. So in the end of this step, for each gene we have the dynamic parameters and the prior that we use to fit them. For the next step, for the clustering, it's very easy. We just remember which prior it was, and we assign the genes to clusters according to the prior that we used. So that all the genes that came from the green cluster, from the green prior, are now in the green cluster, and in the blue cluster, and so on. And since the original clusters were only an initial guess, we now use the new clusters as input for the next stage. So in the end of this stage, we also have cluster assignments. So now that I've shown you how it's done, let's see if it really works. So we need to test the three aspects of our algorithm: the clustering, the modeling, and the estimation parameters. And in order to do that, it would be really great if we had some data where we knew all those, the clustering and the parameters, and we could compare them to the estimated ones from our algorithm, but we don't really get that in biological data. So we need to create some synthetic data. So what we did is actually two things. One way to add noise to data is to add noise to the expression data, which is actually how much the measured expression deviates from the real expression value. And another way is to add noise to the parameter, which is the amount of variation within each cluster, or how much each gene deviates from its prototype within the cluster. So we choose some prototypes from real data that we have, and we create data sets. And to test how well we cluster, we compare ourselves to k-means based on Pearson correlation distance and Euclidean distance. And you see here on the x-axis is the increasing amounts of noise. On the y-axis, it's the mutual information, which is a measure of how well the genes within each cluster fit together. So the higher, the better. And we see that we do more or less the same as the other algorithms. How do we do with modeling? So here we compare ourselves to Chechik et al.'s model, which was the one with the 6 parameters, to our revised model with 7 parameters, but this time just one step of the algorithm, without the fitting to priors, without the clustering, and to our iterative algorithm with the clustering and fitting. And here we measure the fit error, which is how much the estimated value in time points that were not part of the training set, how much the estimated value fits in those time points. So we see that in increasing amounts of noise, we are a lot more robust to increasing amounts of noise, and we tend to less overfit the data, which is what we are aiming for. And same goes for parameter estimation. So here we again compare ourselves to Chechik et al. 6 parameter model, and fitting just one step fitting without priors, and we measure how much the parameters we estimate are different for the real parameters of the data, and we see again that we are more robust to increasing amounts of noise and less overfit. So now we see that it works. How can we use it in real biological data? So we were interested in studying the transcriptional response to pathogen, and a case study is a work done by Amit et al., which measured transcriptional response to stimuli including LPS, which mimics bacterial infection, and poly-IC, which mimics viral infection. And they measured the response in dendritic cells in mice in 9 time points over 24 hours. And you see the expression patterns for the genes where red is high and blue is low, and we get both expression for LPS and poly-IC. So we can later compare between the stimuli. So we chose more than 1,000 genes of their data set that were, these genes were either repressed or induced in either one of the stimuli, and we took both of the expression patterns into our data set and applied our algorithm. So now our data set has each gene is represented in both in the LPS expression pattern and the poly-IC, which will be important for later. So we applied our algorithm, and we got 8 dynamical response prototypes, and maybe you can see that the onset time and offset times are marked on the prototypes, and we can roughly set the threshold of the onset time at 3 hours and get early response prototypes and late response prototypes. So in detail, we have 2 early response prototypes, which differ in their magnitude, 2 late response, which also differ in magnitude, and 1 late response, which is repressed rather than induced, and 2 very late response, which with onset time at 12 hours, and 1 very low response cluster. What kind of enrichments do we find in those clusters? So remember that we also have genes response to LPS and poly-IC in those clusters together. So in the yellow cluster, we get enrichments for inflammation response under LPS and antiviral response under poly-IC, which makes sense. In the light blue, we have transcription regulation and NF-kappa B regulation, I-kappa B alpha. a lot more that I'm not showing here. We also can look for motifs in the clusters, and we found that every cluster was significantly enriched for at least one motif, and you can see some of them here. So we see that we indeed get biologically meaningful and correlated clusters. We can also look at the genes point of view. So remember that we had also LPS and poly-IC response. So LPS is the solid line, and poly-IC is the dashed line, and the colors are according to the clusters in the previous slide. So, for example, we have genes like TNF, which have the same onset time under both stimuli, but different magnitude. Genes like FAP3, which differ in onset time, but not in magnitude. And genes like CXCL10, which have the same response under both stimuli. And this actually defines new groups for us. We can look at all the genes that have one sort of response under LPS and another under poly-IC. So what we see here is for each cluster in LPS, the distribution of its genes in the poly-IC cluster. So, for example, if you look at the yellow cluster in LPS, you see that roughly 30% of the genes there remain in the yellow cluster in poly-IC. So it's the same response. And you see that roughly 10% of the yellow cluster in LPS switch to the light blue in poly-IC, which is like TNF there. And we also see that a lot of the yellow and light blue clusters in LPS, which are the fastest and the highest, switch to the later orange and red clusters in poly-IC. And also that a lot of the genes have the same, of the later genes have the same response in both LPS and poly-IC. So this is a good way to look at the, to compare between stimuli in genes. Another thing we can do is, forget about the clusters now, this is, we looked at the all the gene sets that we can find in our database, and we wanted to see if they are characterized by a specific parameter. So you see the cumulative distribution of the specific gene set in blue against all gene sets in red. So this is from a KS statistical test. We found that cytokine genes have a significantly higher response peak height than all the other genes, and node-like genes had significantly lower onset time. So this is again to say that parameters, dynamical parameters, are a better way to look at the transcriptional response over time. So to sum it all up, I've showed you a method that clusters, models, time course gene expression data. It provides us with biologically relevant dynamical parameters. It avoids overfitting and is robust to noise, and it provides an easy way to compare between experiments. Now, what I would like to add is that if your data, if the specific model doesn't fit your data, you can switch to another model, you can switch to other parameters. This is a general approach for analyzing time course data. This is the most important part. So I would like to thank Neil Friedman and Naomi Habib and all the wonderful people at Neil Friedman's group in Jerusalem for the amazing opportunity to work with them and learn from them, and they've taught me a lot. And thank you.